to this place called the Cueva de la Zouvre. And I was there mostly as a field assistant trying to study how these fish live in complete darkness. And I really got quite intrigued to kind of trying to understand how this really strong selective gradients between a cave and the outside cause phenotypic evolution, which is kind of evidenced in this picture right here. On the right side, you see a cave fish from about 20 meters inside the cave. On the left side, you see a surface fish, which is right at the cave axis. So these fish were collected about 30, maybe 40 meters apart. There's no physical separation, and you can see they're looking quite different. And so I got really interested in kind of these extreme environment and the strong sources of selection, and how that shapes trait evolution in organisms. But while I kind of came for the darkness and was intrigued by the absence of light as a source of selection, ultimately I really stayed in these kind of extremophile systems because of another source of selection. Because the Cueva de la Zouvre is not only dark, it also has very high levels of hydrogen sulfide. And hydrogen sulfide is a very peculiar gas with kind of these conflicting biological effects. Because on the one hand, it serves as an energy source in ecosystems where it's common. So what you see here kind of hanging from the ceiling inside the cave, these are actually microbial colonies that absorb hydrogen sulfide from the air, use it like plants use sunlight to produce energy, and as a byproduct, they produce pure sulfuric acid. And it's kind of this chemoautotrophic primary production that's really the base of the ecosystem in this sort of environment. But then on the other hand, and you can see this by me wearing this gas mask, hydrogen sulfide is also extremely toxic for all organisms that are aerobic. And it's because it interferes with mitochondrial ATP production, and fairly low concentrations of sulfide are actually deadly. And so to me, this was really interesting as a graduate student. It's like, how can these fish survive not only in absolute darkness, but also in presence of these highly toxic chemical. And so over the year, I kind of followed the sulfide outside of the cave, because there's only 2 known caves with hydrogen sulfide in them. Only one has fish. But it turns out there's a lot of surface sulfide springs, and many of those have actually been colonized by fishes. And so about 10 or 15 years ago, I'm ashamed to say it's not really 15 years ago, a lot of our research actually involved really just stomping around southern Mexico, trying to find suitable study systems. So streams like the one you can see here in the picture that are fed by springs containing high concentrations of sulfide, and that are also inhabited by fish, because there's many springs around that don't actually have fish. And so in this process of finding study system, I became quite obsessed with one particular species of fish. This is called Basilia Thermales. It was described in 1863 by an Austrian ichthyologist. And, you know, ever since the first description, we've really not known anything about this species. It was actually considered a synonym to a more widespread species, Basilia mexicana. Nobody really thought this was a real thing. But I was intrigued by this little fish because in the first description, from 150 years ago, it says clearly that the fish was collected in a sulfide spring. The problem was that the locality description was really poor. It only said it was collected from a place called La Esperanza in Chiapas. And if you Google La Esperanza, it means hope in Spanish. And Chiapas, you find many localities. Every hill that has a beautiful outlook seems to be called La Esperanza. And so for many years, we didn't quite know where this fish was really from. And we got distracted by other things. And it was not until later that I was made aware by an autobiography by Karl Heller, who was the son of the botanist to the emperor of Austria. And he was essentially sent out by the emperor and his father to travel the tropics and find beautiful plants for the emperor's palace. And he was one of the first people to kind of rigorously travel and collect in Mexico. And he did so alone. So unlike at the time where many of the great grand expeditions, right, were kind of these big ordeals, Heller traveled all by himself. And he wrote this beautiful autobiography called Reisen in Mexico. And I think it was my one of my students that first, Maura Palacios, who first heard of this book. And when we turned up a copy, the cool thing was that there's actually a chapter about sulfide springs in there. Now that got me really excited, because finally we kind of had suddenly a better idea where La Esperanza actually is. It turns out it was only about 5 miles away from the field station that we have been staying at for years. And it said very clearly that it's the spring is along the highway from this town Teapa to Tuxtla Gutierrez. And we tried to find this place, and we still couldn't find it. And it turns out that the book copy that we had was actually missing the maps that were supposed to be in the book. It's because people, they buy and sell books. They take out the maps, they sell them separately for a lot more money because they're more valuable. And so it was not until I think 2011 or 2012 where we actually got a copy of the maps. And it turns out way back when, 150 years ago, the highway to Tuxtla Gutierrez was on the other side of the river. And so that is now all private land, and it took some effort. But at some point, we were finally at this place that is called La Esperanza. And it was a really, I remember it fondly. It was kind of a big moment for me. Kind of felt this collector connection to Karl Heller, right, who was standing there in 1848, like 160 plus years before us. And I was just really excited to see what Basilia Thermales really looks like. And we, of course, brought some seines and we dipped in. And this is what we pulled out. So if you're familiar with fish and life-bearing fish, this is kind of your typical molly. And I was enthralled. They had that big heads that we know many sulfur piscillates have. They had these lip appendages that are unique to some populations as well. And so why I was super excited, my brother-in-law, who was with me at the time and who was also a fish biologist, said, "Why are you so excited? These look just like all the other stupid mollies that we've been collecting in Sulphur Springs over the past weeks." And he kind of stole my thunder a little bit there, because he was kind of right. They did look like all the others. And so I was probably not the first one to be kind of caught in that, right? Because wherever we go in nature, this is a widespread phenomenon that different biological lineages that are unrelated to one another, they've evolved very similar traits when they're exposed to similar sources of selection, like these two cichlids here, one from Lake Tanganyika in Africa, one from Lake Malawi in Africa. These are very distantly related and yet have evolved very similar body shapes, very similar functional morphologies, very similar color patterns. And there raises, of course, some big questions about the nature of evolution, because on the one hand, we know that contingency is really important in evolutionary change. We know that selection acts on these random mutations that lead to a certain historical contingency of how evolution unfolds. And then on the other hand, we have evidence for determinism where different lineages involve really similar traits. And it turns out this is a really old debate. Probably one of the most fervent supporters of the role of contingency in evolution was Stephen Jay Gould, who kind of famously quipped that replaying the tape of life 1 million times would never give you the same evolutionary outcome. And while at the same time also being said that, of course, this experiment cannot be done. But it turns out gold only represents kind of one continuum of kind of the perspective, because Simon Conway Morris, for example, upon investigating pretty much exactly the same evidence as gold did, concluded that evolution was more predictable, and that evolution is more like a drop of water that kind of follows the pull of gravity down according to a predictable path on an adaptive landscape, ultimately leading to predictable evolutionary outcome. Essentially, Conway Morris argued that there's kind of a finite number of ecological niches, and there's a finite number of engineering solutions that organisms can use to exploit these different niches. So there's very different views, right, about how evolution unfolds. And if Gould's right, then evolution is probably never going to be a predictable science, because contingencies, by definition, are impossible to predict. But if, on the other way, Conway Morris is right, well, then we might be able to find the rules that define this sort of adaptive landscape and make prediction about how evolutionary change unfolds. So over the years, I've become really interested about the role in the role of predictability in evolution. And we've essentially used these sulfide springfish as a model to investigate that, because it turns out there's not just one sulfide springs, there's not just Basilia Thermales in this La Esperanza spring, but in southern Mexico, there's actually these parallel river drainages and that are all part of the Rio Grijalva river basin. And in the foothill of the Sierra Madre de Chiapas, you get in all of these river drainages, you got got them dotted with these sulfide springs, as indicated by these yellow triangles. And in all of these river systems, we see independent colonization of the springs by species of the Basilia mexicana species complex. So what this leads to, right, is that we have kind of multiple evolutionary replicates of closely related populations in sulfide springs and in adjacent non-sulfidic waters. And we can then ask, well, has evolution unfolded in similar ways in these springs? Or has every colonization event led to different evolutionary patterns and mechanisms of adaptation? And so that's essentially what I'd like to talk to you about today. Now, just to give you a little bit more background about these springs, here's an aerial picture of La Esperanza. And I just want to give you a sense of kind of the spatial scale. These springs are really small, invariably. So the spring head of La Esperanza is right up here, and you can see it flows about the length of about 3/4 of a soccer field until it merges with the Ixtapangahoya River down here. So these springs are extremely small habitats. They're actually completely connected to the river. Here's kind of the view on the river bank. You can see the spring flows in here. It merges with the river. And so there's really no physical separation between regular freshwater habitats and these toxic springs. What's really interesting, though, is that despite the lack of physical barriers that will prevent fish migration, we see really strong genetic differentiation between sulfidic and non-sulfidic populations. So when we collect fish right in here, actually this dark cloud here is a group of probably 5 or 10,000 sulfide spring fish. We collect fish in here, and we could collect fish across the side and the other side of the river. We use population genomic analyses to kind of measure the degree of population genetic differentiation. You see very strong levels of differentiation across really small scales. So within dozens or a few hundred meters, you can see that we see very strong population structure. So these are structure plots. Each column essentially is an individual, and color-coded is essentially the probability that an individual belongs to either a sulfidic or a non-sulfidic populations. And you can see the assignment probability for the sulfidic fish is really 100%. They're genetically very distinct. There's only kind of low levels of gene flow coming from the sulfide springs outside of the spring, but not really the other direction. So this is really an opportunity to kind of contrast and compare populations in sulfidic and non-sulfidic waters and study how they are different and how they're actually managing to survive in the toxic environment. Now, in order to do that, we started out to do a lot of kind of population genomic and transcriptomic analyses, and we have very clear predictions of what we might expect in these systems based on kind of a long history of toxicological and biomedical research associated with hydrogen sulfide. Because it turns out we know a lot what this toxic gas is doing inside of the body. We know exactly why it's toxic. The reason why it's toxic is because it shuts down ATP production in mitochondria. So you might remember this from introductory biology here. This is the inner mitochondrial membrane, the respiratory chain. So this is where oxidative phosphorylation happens, right? Essentially, these oxidative phosphorylation proteins, they harvest electrons from organic substrates. In the process, they pump protons across this inner membrane. And it's this proton gradient back here that produces ATP from ADP. The reason why sulfide is toxic is because it binds to this complex 4 cytochrome C oxidase, or COX, off the respiratory chain. So this gets blocked, the electron flow stops. Accordingly, the proton gradient gets disrupted. That's why there's no more ATP. That's why it's so toxic and to such a wide array of species, right? Virtually all animals are highly susceptible to these toxic effects. And what's really interesting, though, is that we all also have the ability to detoxify hydrogen sulfide because it's produced as a side product of cysteine metabolism. And this also happens inside the mitochondria. So we have a series of enzymes. The first step is mediated by an enzyme called SQR. This is sulfide quinone oxidative reductase. It essentially binds hydrogen sulfide and it oxidizes it to non-toxic forms. Interestingly, this detoxification process is actually linked to oxidative phosphorylation because as it binds sulfide, SQR gives in electrons into the respiratory chain. So our hypothesis from the get-go was that that sulfide spring fishes, they probably have modified complex 4 of the respiratory chain, and it kind of became inert to the toxic effects of sulfide, or they maybe have modified SQR to allow for much higher levels of detoxification. And so after, in order to test it, we essentially went out, we collected fish from all of these different river drainages, both from sulfide springs and adjacent non-sulfidic habitats, brought these things back for genomic and transcriptomic sequencing. And to kind of sum this up all in a single slide, I think some of my students might be upset because this was entire dissertations, but we found very strong evidence for selection, not only on complex 4 of the mitochondrial chain, but multiple OXPHOS complexes. And we found very strong transcriptional evidence for upregulation of these detoxification pathways. So this happens fairly consistently across all of the sulfide spring populations we have investigated. So the question, of course, is like, is this kind of signal of selection, does this really lead to the predicted phenotypic changes? So what are really the functional links between the genes, the genetic changes we've documented, and the organismal performance in a relevant ecological context? Because it's easy to look for outliers, and it's easy to kind of weave stories together about how these changes are adaptive, but can we really generate hypotheses about organismal performance based on these genetic changes that we have documented? And this is something my grad student Nick Bartz really tackled. And the first thing Nick did is to actually test the hypothesis that these genetic changes in cytochrome C oxidase really led to differences in the susceptibility to H2S. And so what he essentially did is he extracted mitochondria from liver tissues, and he measured the activity of cytochrome C oxidase, so the toxicity target, as a function of hydrogen sulfide concentrations. And what you see in blue here are the non-adapted, the ancestral populations from fresh waters. You can see that the activity of this enzyme goes down, right? This is why sulfide is so toxic. As there's more sulfide, activity rapidly approaches zero. In contrast, in at least 2 of the populations we investigated, we saw that there's basically a reduction of that toxic effect. What it suggests is that these genetic modifications that we're observing in the toxicity target really make that toxicity target inert to the sulfide. Nick also tested whether these changes in the detoxification enzymes actually matter for the rate of sulfide oxidation. So what you see here on the y-axis is SQR activity, that's the detoxification enzyme, again, as a function of hydrogen sulfide concentrations. And as you can see, sulfidic fish here in yellow have much higher detoxification capability than non-sulfidic fish in blue. Now the question is that like, are these changes sufficient to actually maintain ATP production in the presence of hydrogen sulfide? Well, in order to test that, we again extracted mitochondria and we measured mitochondrial respiration, so the respiration of intact mitochondria in the presence of hydrogen sulfide. And so we did this both for sulfidic and non-sulfidic populations, and we got essentially response curves that kind of stylized look like what you see here on the right-hand side. So in non-sulfidic fish, respiration rate kind of dropped really rapidly and approached zero. Again, ATP production stops because sulfide is toxic, right? While in sulfidic fish, we see the decline much more slowly, and it tends to actually plateau even when sulfide concentrations are much more considerable. Now, to quantify the difference here, what we did is we simply calculated the area underneath these curves as a measurement of sulfide resistance. And what you can see here, in all drainages, sulfidic fish have much higher areas under these curves, so much higher mitochondrial function in presence of sulfide than the closely related population in adjacent non-sulfidic populations. So, and long story short, what we have really shown, right, is that these modifications of the mitochondria allow fish in sulfide springs to continue to produce ATP, and essentially circumnavigate some of these toxic effects that are lethal within a matter of minutes for non-adapted individuals. So, looks like convergence, right? Looks like very similar changes have led to kind of the repeated evolution of sulfide tolerant fish in different river drainages. Now, how did this come about? Because ultimately there's 2 possibilities of how these very similar phenotypes might have arisen in the different sulfide springs. On the one hand, we could have selection on de novo mutations, or we could have selection on standing genetic variation. Let me explain briefly what the difference between these 2 scenarios is. So when we have selection on de novo mutation, we have essentially repeated colonization of different sulfide springs from the same ancestral stock. And upon colonization, there is beautifully adaptive mutations that arise independently in different river drainages, such that the adaptive changes that we observe are unique to each spring, right? Because unique mutations have arisen in the different replicates and then swept to high frequencies in the different environments. When we have selection on standing genetic variation, the assumption is that putatively adaptive alleles that confer an advantage in sulfidic environments are already present in the ancestral populations, although at really low frequency. And so even though colonizations are independent in all of these cases, we see that it might be the same alleles that are recruited over and over and over again in these different replicates to mediate adaptation to the toxic conditions. So which one of these is it? This turns out this is actually relatively easy to test, because what we can do is we can resequence whole genomes of all of these fish. We can chop their genomes in tiny little pieces, right? And then for each piece ask, how are the alleles in the different populations related to one another? If we have selection on de novo mutations, we would expect that sulfide spring alleles are unrelated to one another. If we have selection on standing genetic variation, we would expect that alleles in sulfide spring populations are really closely related and form monophyletic groups. So which one is it? Well, it turns out in our case, it's both. When we look at some of the putatively adaptive mutations in cytochrome C oxidase, that's the toxicity targets, we very clearly see kind of independent evolution of these alleles in different populations. But if we look at the evolution of the detoxification genes, we invariably see that alleles in sulfide spring populations are very closely related. So we see kind of this mix between selection on standing genetic variation and selection on the de novo mutations. And to me, this was all kind of a little bit unsatisfactory. To me, this kind of indicated that while we clearly did have independent colonization of these sulfide springs, a lot of the evolutionary innovation essentially happened once. So yes, we can argue this is convergent evolution because these alleles have repeatedly swept to high frequencies in some of these populations. But at the end of the day, these high level detoxification alleles, they originated once. They didn't originate independently. It's an N of one. It's not an N of the number of colonizations, right? And so I really wanted to know whether, maybe other species that colonize sulfide springs, whether they would find the same adaptive solutions, because clearly we didn't have as much replication, evolutionary independent replication as we really wanted. And so fortunately, sulfide spring fish do not only occur in southern Mexico, nor are all the sulfide spring fish associated with the Basilia mexicana species complex. It turns out sulfide springs are much more widespread. And across the Americas, multiple lineages within this family of fish, the Basilidae, have colonized these highly toxic environments. And these are, even though they're in the same fish family, or these are not really closely related taxa. So if you look at this from a phylogenetic perspective, we essentially again went out and sampled both sulfidic and non-sulfidic lineages across all of these groups. And at the base of the phylogeny, this split here is estimated about 40 to 42 million years ago. So these are very deep divergences, and we now suddenly have evolutionary replicates where selection on standing genetic variation becomes really, really unlikely, right? Because these gene pools have been separated by deep evolutionary time, and they're also in terms of biogeography, they're very distant from one another. And so we again went out, we collected fish, we did a lot of transcriptomic sequencing for this project, and we asked, is there any sort of signal of convergent evolution in gene expression across these lineages? We essentially used phylogenetic comparative analyses to do this, and lo and behold, for roughly 250 genes, we find a significant degree of convergence in terms of the shift of expression as fish colonize extreme environments. So just to help you read this figure, each column essentially represents a gene, each row is a phylogenetic lineage. What you see here on the left-hand side is a similarity tree. So you can see that except for one lineage here from the Dominican Republic, all the sulfide spring lineages cluster meaning that in terms of the expression of these genes, they're most similar to one another. And this contrasts with their phylogenetic relationship, where you can see that the sulfide spring lineages really have originated independently. So what sort of genes are in these convergent evolution cluster here? Well, they're all associated with sulfide detoxification, with aerobic metabolism, with sulfur processing and sulfur excretion. So it's essentially the same genes that popped up in our initial analyses in Basilia mexicana. We see the same sort of genes popping up again in these comparative analyses. So perhaps what this indicates, right, is that evolution might be predictable. So we see predictable changes in genes and physiological pathways that clearly matter in the context of sulfide in all of these lineages. And perhaps the reason why we see a high degree of convergence here is because, you know, there's some serious design constraints. There's only so many ways you can deal with hydrogen sulfide. There's only so many ways you can skin a cat. You can either modify the toxicity target, or you can increase your detoxification. And it's maybe the kind of high level of the constraints, kind of the few molecular targets there that cause these repeated changes across all of these lineages. I know almost for certain that if Stephen Jay Gould would be here listening to me right now, he would probably unmute himself right away and say, bullshit. This is a completely biased interpretation of your results. And he would ask me how much lineage specific changes we're actually ignoring as we come to this conclusion. And the answer to that is a lot. Yes, there's very clear signals of convergent evolution in a bunch of genes, but there's also, in some instances, dozens, in some instances, hundreds of genes that are differentially regulated in each of the sulfide springs, right? And in our field, we often have an inherent confirmation bias, where we look for kind of the shared changes across lineages, and then kind of weave stories about adaptive evolution, while we're ignoring all kind of the unique stuff because it's hard to interpret, right? Is the stuff unique because it reflects some contingencies maybe associated with genetic drift, or are those uniquely differentially regulated genes? Are they also involved in adaptation? So I think this is something that we have to grapple with as a field. And so one of the questions that kind of arose from that is, well, how could we quantify kind of the degree of convergence in a more quantitative nature? So rather than asking, is a gene convergent or not, what is kind of the degree of convergent evolution that we're observing across the transcriptome? And in order to do that, we simply analyzed our data slightly differently. So rather than having all of the sulfide spring lineages in the foreground and the non-sulfidic lineages in the background, we simply looked at how gene expression evolves in our non-sulfidic lineages. And then we pretended that it's always only pairs of sulfidic lineages that colonize these extreme environments. And we did this analysis for all possible pairs of sulfide spring fishes, asking what's the degree of convergence just between these two lineages against the background of gene expression evolution in the non-sulfidic environments. And this kind of gives us a measurement of the degree of convergence across all of these lineages. And it turns out that the degree of convergence is highly variable. Yes, in some lineages, you get very strong degrees of convergence, but in other pairs, the convergence, the degree of convergence is actually much, much lower. It turns out the estimates for the degree of convergence vary over, 2 orders of magnitude. And the question then becomes, well, what explains this variation? Is this really just a byproduct of historical contingencies, drift, and other unpredictable things that happen? Or is some of this variation actually shaped by genetic and ecological factors? So what we then ask is, are there any aspects of genetics or ecology that predict how much convergence we see between particular lineage pairs? And the first hypothesis we tested was to ask whether more closely related lineages would have higher degrees of convergence than more distantly related lineages. The rationale there being, if lineages are closely related, they're more likely to have similar genomic architectures that respond similarly to the same sources of selection. They might even share some genetic variation that selection can act upon. And so if we plot the degree of phylogenetic relatedness, so the phylogenetic distance between lineage pairs, against the degree of convergence in gene expression, you see there's absolutely no relationship. More closely related lineages do not exhibit higher degrees of convergence. And so one of these genetic hypotheses about drivers of convergence clearly does not hold up in our case. So the second hypothesis we then tested is we asked whether the ecological similarity of different sulfide springs that different lineages have evolved in actually predict the degree of convergence. So we not only measured, a wide variety of water chemistry characteristics, but we also included a kind of macro, like a climate scale, macroecological variables. And if we then plot the environmental dissimilarity between lineages and the degree of convergence, we see a very strong relationship. And the way to read this is essentially the degree of convergence is highest when the environments and that the lineages are exposed to are most similar. And so what that essentially indicates is that even though these sulfide springs are really small, this is a different spring I haven't showed you before. This is called like Gloria Springs. You see about, I would say 3/4 of the spring run here. And even though these, these springs are small, right? They're not just characterized by the presence or absence of hydrogen sulfide. It turns out that the selective or the environmental gradients between sulfide springs here and the non-sulfidic habitats downstream, they're much, much more complex. And I recently reread Levin's and Lewontin's Dialectical Biologist, and I stumbled upon this passage from 1985 where they said, for the most part, description and analysis of the environment in evolutionary studies is strikingly naive compared to the understanding of the structure and processes of organisms. And in the age of genomics, I think this gap has only widened, right? We have made great strides by simplifying the environment, sulfidic or not, when the nuances here actually matter greatly. So I think what's important to remember here is that going forward, we cannot just treat all sulfide springs simply as sulfide springs, but we have to start acknowledging that the variation in other water chemistry parameters and variation in also biotic sources of selection matters for the evolution of these sulfide spring lineages. So does acknowledging the complexity of the environment solve our problem of predictability? Well, maybe not. There's probably other limits to predictability. For example, there's these deep chasm in signals of convergence at different levels of organization, where we tend to see relatively high levels of convergence in phenotypic traits that are directly related to organismal function, but we tend to see much lower levels of convergence at the genomic level. And it turns out this is not only true for other systems, it's true in our system too. So we look at genome scans, we actually see very few shared outlier loci that are indicative of consistent selection across different sulfide spring lineages. So what you see here is results from whole genome resequencing for 3 lineages, Basilia mexicana lineages, Pseudocyphophorus and Cyphophorus lineage. These are some of the 3 species that exhibit the highest degrees of convergence at the level of the transcriptome. Yet in terms of shared outlier loci, there are almost none. And none of them are actually related to anything associated with hydrogen sulfide. They're mostly associated with immune function and gene regulation. And the only place where we really kind of see a semi-consistent signal of convergent evolution at the level of the genome are our toxicity targets in the mitochondrial genome. So this is again COX-1 and 3, two of the subunits of the toxicity targets. And you can see across these different lineages, we see kind of multiple convergent amino acid shifts. Again, they're not universal though. There's plenty of lineages that do not have any of these substitutions. And then there's plenty of lineages that have unique substitutions as well. So how do we reconcile that? So how do we reconcile kind of the role of contingency and determinism in evolution? And one way to reconcile these different views that Gould and Simon Conway Morris brought to the table is perhaps that the role of contingency and determinism varies by level of organization. So yes, we see high levels of convergence in phenotypes where essentially the organismal function is directly tied to specific trait configurations. But it might be unique genetic changes that get organisms there, especially in traits like the ones we study, sulfide tolerance. These are highly polygenic traits. And there's documented high redundancy, right, in the genome, such that different changes, different genetic changes might actually cause similar changes in the fitness of organisms. And ultimately, this is nothing really new, right? We have known since Kimura's neutral theory that genome evolution is largely contingent, meaning that drift and other contingent factors play a large role in genome evolution, whereas phenotypes are much more prone to be subject to selection because they're actually visible to selection. And so going forward, I think we would do well in acknowledging some of these complexities. So environments are not as simple as we often pretend. It's not just sulfidic versus non-sulfidic, but even small habitat patches that exert divergent selection. Selection is actually incredibly complex. And I would be lying to you if I told you that it was all about mitochondria in our system, because it turns out the phenotypic changes that happen upon sulfide spring colonization are much more complex as well. We know that the respiratory physiology of our fish changes. They get much larger gills. They get changes in hemoglobin proteins associated with oxygen transport within the body. We see changes in metabolic rates, and that likely is tied to the presence of hypoxia in our system. We see changes in the trophic morphology of our fishes. They get different jaws, different dentitions, change in the gastrointestinal tract morphology and function, and that's likely associated with trophic shifts. They eat different things in sulfide springs. We also see changes in the brain morphology and the sensory ecology of our fish that might be associated with energy limitation. We see changes in pigmentation patterns. We see changes in the reproductive life history of our fish. Non-sulfidic fish make dozens and dozens of tiny babies. Sulfide spring fish make maybe 5 or 6 ginormous babies. And then we also see changes in behaviors where costly behaviors like courtship and aggression tend to be reduced in sulfide springs. These complexities are real, right? And I think a big question for our field is how do we actually embrace these sorts of complexities and admit them into our research, both in terms of how we conceptually see issues in biology and how we tackle them empirically, right? Because the reductionist approach of simplifying things has brought us really far, but we might be missing out on some important lessons about how evolution works by oversimplifying our study systems. And so with this, I'd really like to first acknowledge that the land we're working on is the traditional land of the Soque people, and it includes sites of religious importance. I really appreciate guidance of local people and landowners, especially people of Tapihulapa that have guided our research in the past. Also would like to thank some funding agencies, lots of collaborators. This is really the work of my grad students that I'm fortunate enough to present, including Christine, who I think had to go off her talk, but she did all that work on the trophic ecology about 10 or 12 years ago. But yeah, if there's any time, I would be happy to chat with you and answer any questions.
Great, thanks a lot for that awesome talk. I mean, it was great again, like I remembered. But there were a lot of new aspects to it too. I think last time was more this dark light adaptation too.